the fifth level of organization here is going to be a chromatids formation. As we said that 6 nucleosome in first level of organization, which is made up of nucleosome. In second level of organization, 6 nucleosome can form a solenoid. Then in third level of organization, there is a formation of 6 loops to rosette. As we said, different solenoids can form a one loop. This loop of about 6 loops can form a rosette. Then there is a fourth level of organization, which is coiled coils, which consists of 30 rosettes. Then 30 rosettes can form one coiled coils, which is the fourth level of organization. Then a fifth level of organization is going to consist of 10 coils. 10 coils can form a one chromatid. Then when there is a 2 chromatid, it is known as chromosome. So a 10 coils, which is consists of about 30 rosettes, which means a 30 rosettes can form a 10 coil, one coil. This one coil can form 30 coils, 10 coils. These 10 coils can form one chromatids. So the overall compacted form consists of 10,000 folds. Therefore, a metaphase chromosome, which is an inactive chromosome, condensed stage of chromosome, is going to consist of 10,000 folds in the chromosomes. But a compaction level is not uniform. A chromatid can be in two forms, euchromatin or heterochromatin. Euchromatin means it is going to have a radial loops as 30 nanometer loop. As we said, that in the interface there is no uniform loops. Therefore, euchromatin is a condensed stage of chromatin, which is found during the metaphase, where in the interface there is small level of interval loosening off. It is a tightened form of chromosome and also inert chromosome in the metaphase to be formed. This chromosome can have a radial loop about 30 nm, which is a euchromatin. But heterochromatin is tightly compacted region of chromatin, which is constitutive heterochromatin and facultative heterochromatin. These are the two types. In heterochromatin, the constitutive heterochromatin is permanent heterochromatin, but facultative heterochromatin can interconvert between euchromatin and the heterochromatin. Based on the cell division taken place, it can convert it into euchromatin or heterochromatin. This is a facultative heterochromatin. But euchromatin is going to have a regular interval of 30 nm radial loops, but it does not have a regular loops. Therefore, a one coil consists of 30 rosettes. These one coil of about 10 coils arranged to form a one chromatid. One chromatid consists of 10 coils. Therefore, for the chromosome, there is a 2 coils to be present. Therefore, 2 into 10, 20 coils can form 2 chromatids. The 2 chromatids can form one chromosome. Therefore, it is the fifth level of organization, which is a chromatid organization. As we said at the first, A chromosome compaction in eukaryotic chromosome compaction is very important because it is going to fit into the cell of about 5 μm diameter of nucleus, and it is also involved in packaging for preventing of the damage and to transmit to the daughter cells for the gene expression. These are the very important function for the packaging. As we said, it is the overall view. This is going to be a nucleosome, which is also known as bead and spring model, which consists of a core histone protein, is an octamer, which consists of H3 and H4 at the central, H2A and H2B of dimer flanked on both region and top and bottom, and H3 and H4 are going to be a tetramer at the central. This forms the core histones. These core histones is wounded by core DNA, which is about 146 base pairs. This is going to be a linker DNA, which is going to be 82 114 base pairs. A core histone is going to be consists of 6 nm in height and 11 nm in diameter. These nucleosomes are going to form a second level of organization. It is first level of organization. A DNA is compacted into a nucleosome where the histones are going to be basic and DNA is going to be acidic. Based on its property, it is compacted off. Then it was further compacted in the second level of organization. A main protein to be involved is H1 protein, which is histone 1 protein, is going to compacted off by connecting the core histone and linker histone. Linker and core histone is going to be connected off. Second level of organization can be solenoid or zigzag. But solenoid is going to be a common method where in both it is going to be compacted from 11 nm to 30 nm. Then the solenoid is further compacted into rosette, which is third level of organization. To compact it off to a rosette, it is compacted to 80 nm. So this compaction need between solenoid and the third level of organization rosette, there is a formation of loops. Solenoids are arranged into loops. About 6 loops can form a one rosette into 80 nm. This looping off can be carried out by non-histone proteins, which plays an important role. A DNA which consists of SAR1 region, which causes a non-histone proteins to bind. Then the rosette, which is about consists of 6 loops, are going to be further coiled into coiled coils of about 30 rosettes can arranged into coiled coils, which is about 100 nm. Then these 30 rosettes are going to form a coiled coils. About 10 coils can form one chromatid. Therefore, 20 coils can form 2 chromatids, which is a chromosome. A chromatid is going to be a fifth level of organization. Fourth level of organization is coiled coils. Fifth level of organization is chromatids, which is about 400 nm in diameter. Therefore, it can fit into 5 μm diameter of nucleus. This is the organization of the gene or organization of a DNA. A DNA can form a gene. A gene is going to be formed by chromosome like this level of organization. A 5 level of steps are involved